So today's topic is adipose cells versus bone marrow cells, and I think this is something that is probably heavily debated throughout the space, and it's something that I've spent quite a bit of my time researching. And as many of you know, I began my field in regenerative medicine with bone marrow-derived cells, and for many years worked with these, including working at Osiris Therapeutics. I was responsible and worked with a team working on a product called Chondrogen, which is for osteoarthritis. And these are culture-expanded bone marrow cells that are allogeneic from a non-matched donor. And we were hoping to utilize those to grow new meniscal tissue after a patient had a partial meniscectomy, and saw many great results with those cells. I also worked for Tulane University, where I worked with bone marrow cells for spinal cord injuries, also culture-expanded. And I've now been with BioHeart for 10 years and have worked with about 6 of those years, maybe even a little bit longer, and also working with muscle-derived stem cells. So I have some experience with both cell types, and I think that they can both be beneficial, but today I want to compare them, and we're going to look at several publications that have either been recently published or some that are a couple years old to demonstrate the differences. And I think it was important to look at articles that have done direct comparison between bone marrow and adipose, because adipose are sometimes considered the new kids on the block, in the sense that there are less publications with adipose than there are with bone marrow, and that's mostly because adipose cells have been discovered as having high amounts of mesenchymal cells much later than what bone marrow were. And I think the other thing that is sometimes confusing for people is the word bone in bone marrow tends to make people think that it makes bone better than maybe adipose does, and that's not necessarily the case. So don't necessarily think about the name. Those names were given before there was a full panel of research done on comparing the two different cell types. So before we start with the specific papers, just a quick overview of stem cells. There are stem cells found in every single tissue in your body, and the main purpose of these cells is to repair and maintain the tissue with which they are found. And they can exist in an undifferentiated state and multiply, make copies of themselves, and then differentiate to form new tissue. One thing that's unique about these cells is that they are multi-potential, which means that they have the ability to form many different types of cells and not just one type necessarily. This is slightly different than the embryonic stem cells that we sometimes hear talked about in the media, and those are going to be pluripotential, and they have the ability to form just about any cell type. So it's important to understand those differences, and I think it's also important to communicate those differences to your patients, because oftentimes patients or the general public do not necessarily understand the differences between embryonic stem cells and adult stem cells. So in my case and in my experience and research, I have only dealt with adult stem cells. I have not done anything with embryonic stem cells on patients. The other thing that's unique about stem cells in general is that they exhibit something called plasticity. And what this means is they are easily influenced by their environment. So it doesn't necessarily matter where a stem cell starts or originates, but it matters more where it ends up, because that will dictate what it becomes. And the environment surrounding that cell helps to make it go down a certain pathway, and that's called plasticity. There are a lot of different tissues inside your body that contain stem cells, and we're seeing many publications coming out discussing the different places that you can obtain stem cells. And I think there's also different stages of stem cells. So you have your pluripotential cells, which are going to be your embryonic type cells, but then you also have on the other end of the spectrum your unipotential stem cells, which are those that can only form one type of tissue. So that would be something like as from your skeletal muscle. Those cells can only create muscle. Those are referred to as a unipotential stem cell. They can also be called a myoblast. So there are many different tissues that can allow us to obtain stem cells. In today's talk, we want to talk about bone marrow cells and adipose cells, and in both of these populations there is something called mesenchymal stem cells, and we believe that mesenchymal stem cells can go down a variety of different pathways and form many different types of tissue, things like bone and cartilage, muscle, kidney cells, vessel cells, heart, nervous system, and that even there's a population within these mesenchymal cells that exhibit a hematopoietic type lineage and can help to create some of those blood cells. So oftentimes we talk about two different stem cells being mesenchymal and hematopoietic, but really within that mesenchymal stem cell population, those cells themselves actually have the ability to form some hematopoietic type lineages. These are some of the characteristics of the cells that can be found within the bone marrow. So if you take a bone marrow aspirate, which is going to exist basically in the middle of the bone, you have these different populations of cells that are within there. You have your hematopoietic cells, which are shown at the top. I'll place my arrow here, and these are going to be responsible for forming all of your blood cells. So things like your natural killer cells, your lymphocytes, your macrophages, your platelets, your red blood cells. You also then have these mesenchymal type cells, which are often referred to as a stromal cell or a stromal stem cell, and they have the ability to go down different pathways to form more tissue type, things like bone and cartilage, even skeletal muscle, and then also having this ability to form the hematopoietic stem cells, which would then be responsible for creating the blood. These cells also have the ability to form fat, or here you can see an adipocyte. When we talk about the cells that can be obtained from a stromal vascular fraction, which is from the adipose, these contain many different types of cells as well. So it's a mixed population, whether you're coming from the bone marrow or whether you're coming from the fat. So within the fat, you're going to have many different types of cells, including adipocytes, the fibroblasts, smooth muscle cells, endothelium progenitor cells, but then most importantly, you're going to have this population of the mesenchymal cells, or those marrow stromal cells, as we sometimes call them. This is actually taken from a slide that Cytometry often presents, and Klein-Hens had presented this in May of 2001, showing what can be obtained from this stromal vascular fraction, often called a cell pellet. So things like your hematopoietic stem cells, your endothelium progenitor cells, even your parasites. Now, the parasite is something that's really interesting, and this is something that you may have seen presented over the past couple of years. There's a couple different groups that have discussed the importance of this parasite, and the parasite being the precursor to the mesenchymal stem cell. Now, the parasite is something that coats all of the vasculature inside of our body, so it makes sense that you can get these mesenchymal cells basically anywhere that you can find vascular tissue. So if you remember, an adipose sample is going to contain quite a bit of vasculature, and that's kind of demonstrated and depicted here in the picture on the left of your screen. And those are going to be coated. All of the parasites that would exist in your body can be found on that vasculature. So those can be thought of as the precursor to those mesenchymal stem cells. So then you also have fibroblasts, as well as what we call here adipose stem cells, which that term can be used interchangeably with mesenchymal type stem cells. So this is from another publication in the European Heart Journal. This was actually from a study also completed by Cytometry, demonstrating that the adipose cells could be used point of care in heart patients, and they delivered the cells into the heart. But what they did was test to see the difference between the number of nucleated cells that could be obtained from equal volumes of fat or bone marrow. And what they found is that 500 times more of these cells could be obtained from the adipose tissue than from the bone marrow. So this was pretty early on in 2007, demonstrating really the importance of the adipose tissue and the number of cells that could be obtained from it. So I think a good rule of thumb is that from 60 cc's of adipose tissue to 30 million cells, nucleated cells, and those are going to be part of your stromal vascular fraction. So I always think of it as somewhere between 0.5 to 1 per 1 mil of tissue. So in other words, if you have 1 mil of fat, then you would have 1 million or maybe 500,000 cells. This is from another publication, and what they concluded in this publication is that adipose tissue can be obtained by a less invasive method and in larger quantities than bone marrow. And I think the dilemma with collecting bone marrow is that unless you have an excellent technique that requires you to travel down the length of the bone to ensure that you're constantly pulling bone marrow, you're going to pull quite a bit of peripheral blood instead of whole bone marrow, and I think it becomes difficult to get large volumes of bone marrow. So you're going to be limited in the volume that you can collect. In the case of adipose tissue, most patients have more than enough, and you can collect quite a bit of adipose tissue in a non-invasive method, only local anesthesia, and it doesn't require you to puncture into the bone to collect it. So the first paper that I want to discuss today is called A Comparative Analysis of Mesenchymal Stem Cells from Bone Marrow, Cartilage, and Adipose Tissue. Now, I know the point of today's talk was to compare bone marrow and adipose, but we'll also show the results of this paper, which show the differences in cartilage. And I just think it's important to note that the MSCs can be collected from many different locations, and what exactly are those differences going to be. So the first thing that they looked at in this paper was the expansion characteristics, or the growth speed. And what they noticed is that the adipose MSCs grew at the highest speed and kept almost the same growth speed throughout 10 generations. So if you look at this graph below, you can see the green line is going to be your adipose cells, the black line in the middle is going to be your cartilage cells, and then finally the bone marrow cells at the bottom. So you can see the adipose cells were significantly higher than any of the other two groups. Looking at also the proliferation and the growth capacity and the proportion that were in the S phase in the cell cycle. Now remember, the S phase is actually the growth phase of the cell cycle. So having a longer percentage of time in the S phase would indicate greater proliferation. So when comparing bone marrow, cartilage, and adipose cells, the S phase was the highest for the adipose cells. So if we look at the graph all the way to the right, this section that is right here with the lines going through it, this is going to be your S phase, and this phase was the longest and largest for the adipose cells as compared to cartilage and bone marrow. It had a higher percentage. So you can see the numbers here, 20% versus 5% and 9% for the bone marrow cells. The other thing that they looked at is what were the differences in the telomerase activity. And what they found is that there was no difference between the passages of identical cell type in passage 6. So this is great because it indicates that you're not losing any telomerase activity as you culture expand those cells. And that can be important because sometimes with cell populations, as you culture expand the cells and as you go out in proliferation and passage, you find a reduction in functionality, and this sometimes is linked to the telomerase activity decreasing. So that all of the cells across the board showed similar telomerase activity at passage 3 and passage 6 was important. Now, there were slight differences between bone marrow and adipose, with adipose being slightly higher in telomerase activity, but there was a statistically difference in the cartilage cells versus adipose, as well as cartilage versus bone marrow, both of those being adipose and bone marrow being higher than cartilage. And as I said before, no statistical difference between adipose and bone marrow, but with adipose slightly higher. When looking at flow cytometry, or the proteins that were expressed by these different cells, what was found is that they had very characteristics, the bone marrow cells, the cartilage cells, and the adipose cells. The main differences were between the CD73. The cartilage cells expressed slightly lower than what adipose and bone marrow did, adipose being the highest at 96% versus only 84% for bone marrow, and then of course much lower for the cartilage cells. For CD44 and for CD90, which are traditionally thought of as mesenchymal cells, they were very similar, with the exception of the cartilage cells expressing much lower amounts of CD90. And again, CD90 is thought to be a mesenchymal marker. The other thing that's interesting to note here is that when the cells are culture expanded, the adipose cells do not express CD34. CD34 was traditionally thought of as a hematopoietic stem cell marker. What we've actually discovered is that the stromal vascular fraction, which is the population that is taken off of an immediate separation of the fat tissue, does actually express CD34, but downregulated regulated as the cells go into proliferation and culture expanded, and then it no longer expresses CD34. I point this out because in one of the papers that we're going to present at the end, they actually have differing data. They demonstrate that CD34 continues to be positive, even passages out for adipose-derived stem cells. The other thing that's important about CD34 is that it is thought to be linked to angiogenesis. And so this is why it is believed that the adipose cells can have a higher capability for creating new blood vessels than the bone marrow or certainly the cartilage-derived stem cells. But in any case, the point in this paper is that they were negative. All three cell types were negative for CD34 and CD45, which are thought to be for hematopoietic. They made a summary where they compared the differentiation capability and characteristics of the cells derived from the three different tissue sources. And I think one thing that was important to note here is that the samples from all three different sources were able to proliferate as well as differentiate down the three paths of bone or osteogenesis, fat or adipogenesis, or cartilage chondrogenesis. Now, what was noticed is that some samples had a greater ability to differentiate than others. So this becomes a little bit challenging to measure because differentiation assays are often scored visually. So it's a little bit more of a qualitative test than a quantitative test. And so what they demonstrated is that bone marrow cells actually had higher capacity for osteogenesis than the other two. Adipogenesis was higher for both the adipose and the cartilage, and then for chondrogenesis or cartilage, the cartilage cells were the highest. So I think that probably makes a little bit of sense, but important to note that all three cell types had differentiation potential. And they also demonstrated that all three cell types expressed similar ability for angiogenic type cytokines. So that was no statistical differences demonstrated, and they could not put this in a table to demonstrate any sort of differences. Now, they also did an adipose assay, and they demonstrated that in some cases the adipose cells did better, and in some cases the cartilage cells did better. So it just depended on which assay was being tested there. So the next paper that I wanted to discuss was called Autologous Point-of-Care Cellular Therapies Variably Induce Equine Mesenchymal Stem Cell Migration, Proliferation, and Cytokine Expression. This is a really interesting paper because I think it addresses what is more commonly being used in the clinic. So even though this is done on equine, I think this translates very well to the human side. And they looked at three different samples, and they called these mixed regenerative cell concentrate products. So these are products that are minimally manipulated and can be produced bedside, patient-side, used point of care. The first one is bone marrow mononuclear cells, which they called BMMN. Then they have platelet-rich plasma. And then finally, SVF, or adipose-derived stromal vascular fraction. And what they're trying to determine is the effects of these cell therapies on the mesenchymal cell's function in vitro. So let's look at some of the data. First off, what they looked at was the proliferation ability of those cells in response to one of those three products, either the SVF from fat, the bone marrow mononuclear cells, or they also looked at cord blood cells, and then finally platelets. So they did first a control, which was just going to be baseline proliferation of the MSCs when placed in complete culture media. So this dotted line coming across is the control bar. This is what the MSCs would normally grow if just placed in regular media. They then added one of these four different types to that media to see if it would affect the growth. And what they found was really interesting. The SVF yielded the highest fold change as compared to the other samples. So proliferation was greatly increased and significantly increased when adding the SVF. So the p-value here was 0.008 when looking at the SVF bar. And this is most likely due to all of the different soluble factors that are released in the SVF and all of the different growth factors that are part of that, more so than are part of, for example, bone marrow or even platelets. They also looked at chemotaxis. So this was, chemotaxis is something that is movement because of a chemical stimulus. So what they did is they looked at how the MSCs would migrate toward one of these MRC products, whether it be SVF, bone marrow cells, cord blood, or platelets. And they did this by putting a transwell insert at the end to see if the MSCs would migrate towards it. So they soaked those transwell inserts in either SVF, bone marrow cells, cord blood, or platelets, and the control was just a regular media. So this was just baseline. This is what the cells would migrate this dotted line across if just put in regular media. What was really interesting is they're seeing the same type of thing, that when the SVF is incorporated, you see much better chemotaxis and MSCs towards that transwell insert. They did another experiment to conclude whether or not this was consistent, and they did what they called chemo invasion. And this time they did a very similar thing where they used the transwell insert, but they also coated the transwell insert with a matrigel. And a similar phenomenon was seen where the SVF traveled the fastest, and you saw the biggest fold change as compared to the others. So one thing that was also interesting is that platelets and cord blood cells also decreased that chemo invasion, and that was also seen here in the chemotaxis. They also measured IL-6, or interleukin-6. So interleukin-6 is thought to be both a pro-inflammatory cytokine as well as an anti-inflammatory myokine, and it's thought to be an important part of the process when you're healing. So expression of this could be very important to promoting healing. So they made 3 groups. The first group where they used mesenchymal stem cells by themselves. So this would be represented where my arrow is of the first bar. The second group would be the MRC by itself, so either SVF, bone marrow cells, cord blood cells, or platelets. And then the third group would be the mesenchymal cells plus the MRCs combined to see what the expression would be. So they would do a co-culture. What was very interesting is that SVF was the only one that expressed IL-6 when used by itself. So if we look at the middle bar for each one, SVF was the only one that showed expression of IL-6. When doing a combination, which would be the MRC plus the MSCs, both the SVF and the cord blood showed expression of IL-6 when combined. But interesting that the cord blood cells did not show expression when used by themselves. However, when combined with the MSCs, it did. Both the bone marrow cells and the platelets did not show expression of IL-6. When looking at the fold change, which would be the comparison, if I go back to this graph, the comparison of this bar to the middle bar, the fold change, you can see that, of course, the cord blood had a very high fold change, and the SVF second, and then basically no fold change with the bone marrow cells and the platelets. They also did a similar experiment looking at TGF-beta. Remember that TGF-beta controls proliferation and differentiation of cells, so it would be important to have high expression of TGF-beta when you're doing stem cell treatments because you could get more proliferation of your stem cells as well as better differentiation of your stem cells. So this was really interesting because what they found is that the one that expressed the highest amount of TGF-beta was the platelets. So this actually indicates that it becomes very important to combine your platelets with some of your stem cells because then you can get that high TGF-beta, and I think that's consistent with what most people in the field are saying. You can think of platelet-rich plasma as your fertilizer for your seeds, your stem cells being your seeds. So if you thought maybe just put plasma by itself would be beneficial, what you would be missing out is from the previous graph, the IL-6. But by combining the platelets with the stem cells, you now have high expression of TGF-beta and high expression of IL-6. So there was actually very low expression with both the SVF bone marrow cells and a little bit with the cord blood, but not nearly as high as what was seen with the platelets. This is showing the fold change, so similar comparing that third bar to the second bar, and that's pretty similar across the board. The last thing they measured was prostaglandin E3, and this is the hormone that is involved in that paracrine effect that we often talk about when we talk about using cellular therapy or regenerative medicine, this important paracrine effect. And what was demonstrated here is that SVF is actually the only one that expressed prostaglandin when used by itself. So if you compare this middle bar to the middle bar for bone marrow cells, cord blood cells, and platelets, they do not express it. SVF is the only one. Now, when doing a combination, SVF plus the MSCs, you can see a little bit higher expression. Bone marrow cells plus the MSCs, you can see a little bit of expression, and same thing with cord blood and platelets. But SVF has a much higher expression of the prostaglandin. So the last paper that I wanted to discuss today is adipose tissue-derived multipotent stromal cells have a higher immunomodulatory capacity than their bone marrow-derived counterparts. This was published in 2013. So the first thing they looked at was flow cytometry, and this is what I was talking about before in the first paper where we discussed this concept of CD34 thought to be expressed only on hematopoietic cells. Well, what we know is that this is transiently expressed on the stromal vascular fraction, and what I believe is that that actually gets downregulated once the cells go into culture expansion. However, in this paper, they were measuring some culture expanded cells, and they did find some expression of CD34 that remained once it went into culture expansion. So I suppose it's possible that you could have a small amount of CD34 in adherent cells. Those are cells that will attach. And this would also be an advantage to the adipose tissue because it's thought that that CD34 is responsible for that angiogenesis, and that's something that is not expressed on the bone marrow cells once they go into culture expansion. All of the other markers are very similar. So you can see here CD73, CD90, CD105, very similar to what you see in the row for the adipose tissue, and same thing for the bottom. This bottom row is thought to be mostly hematopoietic type markers, and they're negative for the most part. This is showing the percent positive comparing bone marrow to adipose, and I apologize for the blurry. This paper has a very bad resolution in the pictures, so once you blow them up for slides, they become blurry. But basically, this shows exactly what we were just looking at in those histograms on a bar chart showing the comparison between the bone marrow cells and the adipose cells having very similar expression, with the exception of this CD34, and there was a statistical difference showing that the adipose cells expressed a slightly higher amount of the CD34. This shows the differentiation capacity of the cells to go down the osteogenesis pathway as well as the adipogenesis, and cells from both bone marrow and adipose had very similar characteristics. This is a really interesting experiment. What they did was they placed the cells from either bone marrow or from adipose and co-cultured them with peripheral blood mononuclear cells, or monocytes. And they wanted to see if the combination of the MSCs from either one of those could suppress the growth of the monocyte proliferation. And what was really interesting is that the adipose cells actually suppressed the growth at a higher rate than the bone marrow cells. So this is the control. This is what monocytes look like if you just grow them in culture. So you have 100% proliferation shown here in the black bar. And these are at different concentrations of 1 to 100 to the monocytes, and you can see that the adipose cells actually suppress that monocyte growth better than the bone marrow cells. So how that translates clinically is that the adipose cells can actually help to reduce inflammation and promote healing better than the bone marrow cells if you were looking at this graph. And that's pretty much across the board at all of the different concentrations. The other thing that they measured was the cytokine concentrations, looking at 3 different cytokines. In this case, these are all cytokines that are pro-inflammatory, and what they discovered is that the adipose cells will reduce that cytokine expression better than the bone marrow cells. So looking at IL-12, TNF-alpha, and interferon gamma. All of those were suppressed better by using adipose cells as compared to bone marrow, and that's across the board. So black bar is going to be adipose versus the gray bar is going to be your bone marrow, and of course the first bar shown in each graph is your control. They also looked at something called IDO expression. So IDO upregulation and expression by mesenchymal cells is important because it's the mechanism for the mesenchymal cells to induce the suppression of the monocytes or the mononuclear cells. So the ability to have higher levels of IDO expression means that you're going to have less inflammation. The adipose cells certainly express higher amounts of IDO at both 4-hour, 8-hour, and even the 24-hour time point as compared to the bone marrow cells. This next graph demonstrates whether or not the cells have the ability to inhibit the differentiation of monocytes to dendritic cells. So again, determining whether or not it can suppress an overreactive type immune response. So if we look at the control, this would be monocytes differentiating to dendritic cells. You're going to have very high expression of CD1. So this square right here means high expression of CD1 and low of CD14. So 0.01% expression of CD14 versus 91% expression of CD1. When combined with bone marrow cells, you begin to see that some of those cells that are here are migrating up here and expressing CD14 instead, and you have much lower expression of that CD1. And then finally, when adding the adipose cells, you have very low expression of the CD1 cells and much higher expression of CD14. Looking at B down at the bottom left, this is showing that same thing in bar chart, basically showing that the adipose cells are creating higher expression of CD14 and lower expression of CD1, and they're doing it at a much better rate than what the bone marrow cells are doing. And then finally, looking at different concentrations for both adipose and bone marrow. Adipose is shown here, so you can see that it's having a higher percentage expression of CD14, but also that there is a dose response associated with this, and it has to do with the percentage of cells that are added for the MSCs to the mononuclear cells. So that indicates that there most likely is a dose response when dosing patients. However, at this point, it's very difficult to determine what that dose response is going to be. And I wish I had an answer to, is it better to collect 200 cc's of fat and put that to a patient, or is it better to collect 60? I just don't know. I don't know yet, and it becomes very challenging to do dose response, but we do know that there is some sort of dose response, but where we are on that dose response curve and whether 60 cc's collection of fat is enough, I'm not sure yet. What I can tell you is that it seems to be working. We're getting a response in patients, and we're collecting 60 cc's of fat right now. So we have to just keep studying that, and hopefully we'll know more over the coming years. Next, what they measured was IL-10 expression. IL-10 is an anti-inflammatory cytokine, so higher expression of IL-10 would be better for patients. So in this case, this was the control, and this was the bone marrow cells. And then finally the adipose cells. So you can see the adipose cells had much higher expression of IL-10, and there was a statistically significant difference there. They also looked for cytokine expression of a variety of different cytokines. These are different cytokines that would help with the healing process and promoting anti-inflammatory and pro-regenerative, things like this. And pretty much across the board, the adipose cells had higher expression than the bone marrow cells. So you can see the adipose shown in black is higher for just about all of these, in some cases showing statistical significance, with the exception of VEGF, and there was no statistical difference between these two. They had similar expression profile. Now, to confirm whether or not this was going to be true on a transcription level, they did two more assays. The first one was looking at the mRNA of the interleukin-6 expression, and in fact, there was significantly higher expression of the adipose cells than the bone marrow cells for IL-6, and then also looking at the metabolic activity using what's called an MTT assay, and again, the adipose cells did demonstrate higher metabolic activity with statistical significance. So that concludes the three papers that I wanted to discuss, and basically I think those three papers kind of demonstrate that adipose tissue is the preferred source, And certainly if you have a patient that has adipose tissue, that would be what you would prefer to collect to treat the patient with. Now, a couple things. I wanted to share with you what our current protocols are. First off, if we're going to be doing an IV for a patient, an IV delivery of an SVF, what we would do was place that SVF in saline, and we put this in about 5 cc's of saline, and we'll do a bolus push. I think that it is important not to put this in a bag to drip. The reason being that the cells that are of interest, which are the mesenchymal cells that exist within the stromal vascular fraction, are actually adherent cells. So if you put these in a bag and do a drip that could take 30 minutes to an hour, you're now going to lose some of your cells because they will begin to attach to the polypropylene of the bag. So we don't recommend that. Now remember, if you're going to be doing a bolus push and you're going to be placing those directly into the vein, it's important that you always do a filtration process. So if you're using a kit to collect your SVF, make sure that a filtration process or a cell strainer is part of that process so that you don't create an embolism in your patient. Also, a lot of people ask me, should we be putting platelet-rich plasma with the cells if we're going to be doing an IV? And my response to that is it doesn't make sense. Don't bother. There's no reason to take platelets out of your arm to put platelets right back into your arm. So you may as well just put the cells in saline, save yourself the time and money, and put them right back in. Now, if you're going to be doing a joint injection, an intramuscular injection, intradiscal, intravitreal, or basically anything that doesn't go into the circulating blood, it does make sense to use the platelet-rich plasma and combine that with the SVF. In that case, what we will typically do is get about 5 cc's of PRP. We'll mix that with the SVF and then administer it to the appropriate joint. Now, if we're doing a disc, you're talking about something that's going to be much smaller volume, 1 cc, or if we're doing into the eye, intravitreal, we're talking about something that's very small, only about 0.2 or 200 microliters. So, important to adjust your volume, but keep your same stromal vascular fraction. Remember, the stromal vascular fraction pellet is very, very tiny. It only takes up about 0.1 mil, or about 100 microliters. If your pellet is much bigger than that, it's probably not a very good pellet. You probably have quite a bit of liposomes or other things that are part of that population that you don't necessarily want. So a small pellet is better, and that's a good thing. The other thing that we're typically doing for most of our patients is repeat dosing, and I think this is something that has become critical, especially when we're dealing with patients that have chronic diseases that are difficult to treat. Being able to come back and do repeat dosing for those patients without having to go under lipoaspirate procedure is very important. So what we've been doing is we do that initial lipo, we take some of that fat tissue, we send it to the tissue bank here in Florida, we culture expand, we bank those cells, we can then administer repeat doses for those patients as they're required. And then finally, we will do bone marrow on a patient, particularly if they're just too thin for us to get any stem cells. But in the case of us doing bone marrow, we almost always recommend that they would do culture expansion because we don't believe that that initial bone marrow aspirate is going to be enough to reverse some of the effects of different degenerative diseases or maybe even injuries. It's going to become critical to go back and do repeat dosing. Feel free to email myself. I'm Kristen Camella. My email is there, or Carissa, who helps with a lot of these different webinars that we put on and can also provide additional information about some of the different products that we're offering and banking techniques,